Now,
Tom, I advise everyone that we are recording, and it means that you agree on that by participating in this talk, in the seminar. So it's my big pleasure today to introduce our speaker, Thomas Truskett, who is a professor of chemical engineering at the University of Texas at Austin. It's a big pleasure and also an honor to have him with us. And I would like to use this opportunity also to thank him for supporting our proposal recent, which was accepted by the Foundation for Science and Technology, and his support was valuable, and we would like to thank him. So Tom is an expert in computer modeling of self-assemblies of metal systems, and in particular by applying learning techniques. And today he is going to speak about colloidal nanocrystal gels from thermodynamic principles. So Thomas, please.
First, let me just begin by saying thanks very much for the invitation to participate in this CFTC weekly seminar series. It's a real honor to be able to talk about some of our recent work. I would like to congratulate everyone here on the funding of this new center. It's exciting to hear about it, and I'm looking forward to learning about the fruits of this collaborative work that will be coming out in the next few years. And also would like to say that I'm very much missing being in Portugal today. It would be wonderful to do that at some time in the future, so I'm sure we'll figure out a way to have that. But in the meantime, I'm just thrilled to have the attention of all of these experts in soft matter and in self-assembly. And today I just want to share a little bit about some of the work that our group has been involved with, which is part of a center that we have in Austin, which is funded by the National Science Foundation. It's actually a MERSEC, and the MERSEC focuses on control and dynamics of materials. And the part of the MERSEC that we've been most involved in is really interested in the question of how do we design and realize self-assembly of nanocrystal materials, and in particular gels. And we're particularly interested in understanding how to not only design the structure and the thermodynamic or equilibrium properties of these gels, but also their kinetic behavior, and try to create more broadly platforms for realizing self-assembly. So I mentioned the term nanocrystal here. Probably many or most of you know what a nanocrystal is. Just colloquially, it's a nano-sized particle with an inorganic core, which has a crystalline structure and typically is passivated by organic ligands, which facilitate the solubility of those nanoparticles, but also help to mediate their interactions. So I mentioned that this is part of a team, so I just want to spend a couple of minutes on an acknowledgement here. There's a group of researchers in my lab. Probably the most prominent one on the topics that I'll be mentioning today is Mike Howard, who recently finished a postdoc with us and now is a new assistant professor at Auburn University. Zach Sherman, Beth Lindquist, Ryan Jadrich, and an undergraduate student, Aditya Srinivasan. We work together very closely with an experimental group that's led by Professor Delia Milliron. Their group is really responsible for not only bringing expertise in colloidal nanocrystal material synthesis, but also characterization. Broadly speaking, how do you integrate nanocrystals in materials and then understand the impacts that they have? So many outstanding students, PhD students in her group, some that are still in the group and some that have moved on to industrial positions since the collaboration started. And the third group is Eric Anslin's group. Eric is a physical organic chemist. He's brought his expertise in dynamic covalent bonding and linker chemistry. So in designing the molecules that link the nanocrystals together in solution. And so his students and postdocs have been involved in this project as well. I mentioned this is a project that's funded primarily by the National Science Foundation. We've been lucky in Texas for many years to have funding from the Welch Foundation, which funds fundamental research in chemistry. And we also get funding from the Texas Advanced Computing Center and a postdoc of mine is funded on the Beckman Foundation. So I mentioned that we're interested in self-assembly of nanocrystals, and so this is an area that has received attention for the better part of a quarter of a century, going back to the very first papers where nanocrystals were synthesized. Here I'm showing an example of nanocrystals that were synthesized and assembled into densely packed superlattices. So these are lattices of the nanocrystals themselves. Because you're able to synthesize nanocrystals with such high uniformity in terms of their diameter, they pack into the crystal structures that you would predict, typically very closely packed structures, so FCC crystals, occasionally a body-centered cubic crystal, and those of course been characterized. And if you want to get beyond that, you really need to introduce in superlattice assembly another component, so a second nanocrystal, for example. And then of course there are rules based on very basic physics and packing arguments that will tell you what crystal structures would form for a given composition and size ratio of nanocrystals, and that's really been explored extensively now. So although nanocrystals, when they form superlattices, are sort of the central field of assembly, they get much of the attention in the field, there are some drawbacks to superlattices. So the superlattices only allow you to access discrete geometries. So you can't sort of continuously tune structural properties like coordination number, etc. There's no obvious ways to introduce void space into structures. And of course, many of these systems, once prepared, after evaporation of the solvent are stuck in place. And so the idea of realizing switchable structures with nanocrystals is a somewhat limited one. But there are other opportunities in assembly with nanocrystals which are beginning to receive more attention, which are, I would say, sort of even more directly linked to the soft matter community, and that's nanocrystal gels. And so here I'm just showing you an example of one of the early efforts to make nanocrystal gels from Stephanie Brock's group. Here, these gels were made by chemically removing the stabilizing ligands, which of course leads these particles to be attracted to one another, and so they begin to irreversibly aggregate. And so that's interesting from the perspective that you can form lower density, disordered assemblies of nanocrystals, and of course the structural properties of the assemblies affect their physical properties, their optical properties, etc. So one drawback of this is that because it's a non-equilibrium self-assembly method, you have not as much control over the structure, and then of course the properties that are related to the structure. You can design realized gels that have interesting properties and that retain the properties that are inherent to the nanocrystals that are the building blocks. So here's an example from Eichmüller's group, who has really been a leader in this area. And they have shown that you can form nanocrystal gels. Here I'm showing quantum dots. And the gels actually have optical properties that are shifted relative to the nanocrystal dispersions. Here you're seeing photoluminescence, which is a property of quantum dots, and the PL intensity is shifted because of the coupling that you get, because of the structure that you realize once the gelation has occurred. And of course, people have begun to think about how do you develop systems with tunable properties that have catalytic applications, electronic applications, etc. And so broadly speaking, we would like to be able to think about a platform for being able to do this in a controlled way. In the area of plasmonic nanocrystals, one typically thinks of metals. There have been metal nanocrystal gels synthesized. One drawback with these metal systems which exhibit a localized surface plasmon resonance, or LSPR, is they tend to fuse when they form gels. And so what happens is that the fusion of the nanocrystals themselves essentially eliminates the inherent LSPR properties and any sort of coupling that you might want to design because of the the structure of the gel itself. And so we'd like to be able to look at systems with plasmonic properties in the gel form, where the gel has structure and properties that we control. And so the building blocks that I'll talk about in today's presentation are nanocrystals that are synthesized in Delia Mill irons group and understood by their laboratory's characterization techniques. They're essentially indium oxide nanocrystals with various doping. These are really workhorses in terms of building blocks for assembly because they have shapes and sizes that can be tuned through their synthesis, but also you can tune the absorption via doping levels in the nanocrystals. And you can also imagine if you have a platform for assembling these, you can take advantage of IR hotspots and potentially even switchable materials that have these as their basis. So everything I mentioned will be referring to, in terms of the experiments, these indium oxide nanocrystals. Okay, here's a diagram that I think is helpful for thinking about how to use equilibrium principles to control assembly. I know many of you have seen these concepts probably before in the context of patchy particles. Here I'm showing the phase behavior on sort of this axis is temperature divided by the bond strength, or the physical bond strength between the nanocrystals. On the x-axis is the volume fraction of the nanocrystals. And if we focus on this top diagram, this is sort of the normal gelation picture, where if you create a system that is dispersed and then you quench it into this two-phase region beyond the spinodal, where the bond strength wants to induce phase separation, you'll initiate a non-equilibrium process. So coarsening will begin, and that non-equilibrium process can get locked into a structure which is your final gel structure. And of course, here I'm showing micron scale colloids that have been prepared by this type of process. This is Peter Liu's experiments from 2008. So the nice part about that is it's a way to form gels with pretty interesting ramified open structures. Downside, of course, is that because they're non-equilibrium, they can age over time. They will age over time to dense aggregates, which may ultimately precipitate even on the time scales that you're interested in studying them. So one of the exciting developments that came from many groups, I say sort of principally out of Europe, Francisco Shortino and many others have been involved in thinking about the theory of these, is making equilibrium gels by limiting the valence of the colloidal particles. And that's sort of this bottom picture here. And so the idea is that if you can compress this two-phase region to lower particle volume fractions by restricting the valence of the particles is sort of the most popular strategy, then you can access a region of the phase diagram which has at least closer to equilibrium gel-like properties, and that might not only allow for tunability of gel's properties, but also gets rid of some of the problems that you have with aging, et cetera, in such systems. And so the idea of like, well, can we take advantage of this for colloidal assembly? And of course, I mentioned already that patchy particles is one way in which people have sought to take advantage of such a conceptual picture. So the idea illustrated here on this phase diagram is on this axis, we have temperature divided by bonding strength. On this axis, again, volume fraction. And so if you, over here to the right of this two-phase region, you might think of as a place where you might possibly form a gel. And so the idea here is you change the number of patches that are on your particle, say from 5 to 4 to 3 to 2, et cetera, then you're changing the phase diagram, and in particular, compressing it to lower volume fractions so that you can access sort of an equilibrium gel. So this type of conceptual picture has motivated experiments. So in this area, so DNA tetramers are one example of an experimental system which has a patchy character in the interactions, which could even be programmable, which is somewhat exciting. Of course, you can also think about functionalizing colloidal particles, at least at the micron scale with patches. that have DNA-mediated interactions, and even natural systems like clay particles can have patchy interactions. So this is a concept that has been, I would say, both explored fairly extensively and also understood in the case of micron scale colloidal particles, where it's possible to easily synthesize patchy interactions. But the tools that you would use to do that are much more difficult to apply at the nanoscale. And so there's a motivation actually to look to more macroscopic means for controlling the valence. So I'm going to show you another micron scale colloid experiment to try to motivate this. But the idea here, so this is an experiment that has polystyrene colloidal particles, which are dispersed, and then polymers added, which is attracted to those colloids. Here we're looking at PandyPam, but there are other examples for molecules that could mediate this gel assembly. But the idea is that the colloids begin as a dispersion, and then if you add the linker particles or change the linker to colloid ratio gamma, you start getting limited valence. So you form sort of what we would call bridging gel. And then if you keep adding those polymers, they'll eventually saturate the surface. And so there's no longer opportunities for bridging because you've displaced all of those, and then you have a dispersion again. So it's sort of a reentrant phenomena that has been seen experimentally and is understood conceptually from this picture. And so the idea is that that could be, that sort of strategy could be applied at the nanoscale. And so to understand theoretically and make predictions about that kind of strategy, we can turn to Wertheim theory, which is a theory for association of molecules, colloids, etc. And so I'm going to begin by talking about sort of a very basic version of Wertheim theory, where the predictions rest on the assumption of one bond per patch on a particle. So you have patchy particles, and those particles can only bond once for each patch. In the theory, it treats those patches as uncorrelated. It also assumes a tree-like structure of the network, so no closed loops, and also one bond between a particle and a linker. And just some terminology here, because it's useful for looking at future graphs, like number density, the normal packing fraction of colloids. Here I'm looking at the concentration ratio of the linker to the colloidal particle, and then temperature divided by the bonding strength. So one thing you can do immediately with this theory is make a comparison between a system of patchy colloids, which has microscopic control of valence, and then a linker system, which offers what I would say more macroscopic or statistical valence restrictions. So here's the patchy colloid picture that was published back in 2006 in PRL, which shows the behavior that I mentioned a few minutes ago. And here's a paper that we published in 2016, which explores the system for a binary colloidal system where some of the particles are linkers and the other particles are the building blocks of the colloids themselves. And the main point I want you to get from this plot is that in analogy to decreasing the number of patches per colloid, which you could imagine easily doing experimentally for big enough colloids, you can synthesize particles with different numbers of patches. In the cases of nanoscale particles, you might rather go for statistical control or macroscopic control of valence by controlling not the number of patches per colloid, but the number of linkers per colloid. And similarly, as you decrease that number of linkers per colloid, you compress the phase transition to lower and lower packing fractions and ultimately open up this wide region of phase space for forming open gels or fluids with these systems. So that's the analogy that we want to exploit in this work. Of course, that was sort of a very simplistic model where we're treating the linkers as looking just like the building blocks themselves. For most experimental systems of interest, we'd want to consider linker molecules that look like molecules, not colloids. So we built this also toy model, but one that I think is a little bit more useful for asking questions about these systems. Here we have red red particles, which will represent our model nanocrystals. They will have 8 patches each on them, arranged in an octahedral shape, so sort of symmetric patching interaction. But those patches don't interact with patches on other colloids. They only interact with linker molecules. And so the way that these nanocrystals will assemble in this model is through these bifunctional linkers. And of course, the properties of these linkers will affect not only the structure of the resulting assembly, but also properties that are related to structure. And so the idea is that we could build this toy model and start to understand some of the physics behind what controls structure, phase behavior, and then ultimately properties in these systems. So here I'm just showing you some predictions from that model. So on the left-hand side, these are phase diagrams. The first one is, again, in units of energy. So up here is where the particles are bonded. Here's where they're not bonded, or the thermal energy dominates. And then on the sort of x-axis, this is the nanocrystal packing fraction. And so the case here on the left is for linkers that have molecular weight 2. So it's 2 beads. The beads in this case are like 1/5 the size of a colloid. And then over here, we have the same thing where we're plotting an example phase diagram where the linkers have molecular weight 8, so longer linkers. And in both cases, we have, 1.5 linkers per colloid molecule in the sample. And so the idea here is that the linker, so the phase diagrams look qualitatively the same. The shading indicates the molecular simulation. Actually, the structure factor is 0 wave vector. So this is picking up on when phase separation would occur in molecular simulations. And the curves are actually the predictions from Wertheim theory. And so 2 points I just want to make here. First is that the linker length sets the minimum gel density in the system. So you can think of the length of the linking molecules as being how you control where this right-hand side of the phase envelope occurs. And so longer linkers can get you lower density or more open gels. The second thing is that Wertheim theory does a pretty good job qualitatively of picking up where the simulated phase behavior or phase transition occurs, with the following caveat. The theory is over predicting the effect of linking because it neglects self-linking. So whenever a linker bends back and bonds to itself, like is shown here, it also neglects these kinds of redundant bonds that come about because of bridging. And so I plotted exactly those quantities here. So the dashed line is the case of the fraction, or it tells you the fraction of molecules that are showing you loops. And then the dark lines are showing the fraction of links that are basically wasted by either a loop or a bridge. For the 2 systems here, the blue is for the longer linker system, the red for the short linker system. And these are 2 different volume fractions. But I guess the main point I think you should take away from this is as you increase bonding energy, so try to go towards gelation, you underpredict the fact that you're going to waste bonds in this strategy because you end up getting self-looping interaction and also redundant bonds between colloids. So we'll come back to that point a little bit later. So although you can sort of pick discrete linker molecular weights to tune the density of the gel, you can also think of other strategy like using mixed linkers to get the best of both worlds. So here I show a mixture of short linkers and longer linkers, so 2 and 8, which would give you an average of 5. So here's the phase diagram for a linker system with the mixed linkers, and I'm comparing it to the phase diagram of a colloid linker system where all the linkers have molecular weight 5. And so there's sort of an interesting thing that you can observe here. One is that when you use a mixture of linkers, you actually get phase behavior that's not that different from the case where all of the linkers have the molecular weight of the average. So you can introduce flexibility to the system in terms of local structure, which I'll get to in a second, without compromising the phase diagram. So mixtures preserve the phase diagram, but then when you look at the local structure, what you find out is that while linkers that have length 5 never bring particles, or very rarely bring particles close enough together to realize nearest neighbor interactions that are important for example, hotspots or coupling for LSPR interactions, the mixed linker case will have some finite and controllable fraction of such nearest neighbors that will be present. So you can increase the number of near contacts, which is shown by blue particles in this comparison of short linker, long linker, and mixtures without changing the phase behavior much. So it's really just another handle by which you can tune structure while keeping phase behavior the same. Okay, so I want to start moving toward experiments based on that sort of background from a foundational perspective. And this is where Eric Anslin's group comes in. I mentioned that their group has helped to design linkers for these colloid linker systems. And what I'm showing you here are 5 different pairs of tunable, orthogonal, reversible, covalent bonds. And so that really these are reversible bonding interactions that allow you to have programmability of your linkers. So experimentally, we'll focus on some results here in a second involving this pair, which has an hydrazide aldehyde bonding interaction between the functionalized ligand and the linking molecule, which will allow for the colloids to be driven by this reversible covalent interaction. And then, of course, the assembly can be reversed by turning the chemical reaction the other way. And the nice part of having these 5 orthogonal interactions is that in the future, you could build in programmability of assembly of differently functionalized colloidal crystals. So it really gives you some flexibility. And so here's a cartoon of the linker molecule plugging in and driving the assembly. If you want to sort of think more broadly in terms of this as a platform, you can imagine having 2 different nanocrystals as a simple extension where now you have differently functionalized nanocrystal surfaces, and of course different linkers would connect those together. And so you could exert macroscopic control on the assembly process or statistical control just by the compositions of the linkers that you add to the mixture. So here's the experimental system that we've recently published on in this area. So we take advantage, as I mentioned, of this aldehyde hydrazide dynamic reversible covalent bonding. The linker that was designed for the system shown in the middle, this yellow sort of female addition of the plug, or version of the plug. It's a difunctional molecule with the hydrazide ends on both sides, and of course one of them would connect to each end of linkers that are functionalized to the colloid. So here I'm showing you what the linker actually looks like. So it's got a tridentate strongly bonding surface group, part of the chain that was designed to make the nanocrystals soluble, and then a what I call torque binding group, which is essentially presenting the aldehyde into the solution, so providing this sort of male counterpart to the linking interaction. Okay, so here's some just results from experiments. On the left-hand side, micrographs. On the right-hand side, structure factors coming from small angle X-ray scattering. The idea is that low linker concentrations, you have a flowing clear liquid, and then you add linker and the liquid gels. So it becomes solid-like in its mechanical properties. You can turn the vial over and it doesn't spill out, and you start to see a linking structure form. This is, of course, post-processing. This is in situ, what happens to the structure factor as a function of the linker to colloid ratio as you go from none, and then the addition of it from days to weeks. And you can see a characteristic of this is actually a fairly slow forming gel, but that could be modified by choosing different bonding chemistries. And if you want to think about this more conceptually, go back to those phase diagrams I mentioned before. Here in the middle, I'm showing you predicted phase diagrams from the naive Wertheim theory, but applied with molecular linkers. And what you see is, compared to the experiments, here we go, adding linker at a constant colloid volume fraction, and then eventually you do get to gel formation, which you would expect in the two-phase region. But one thing to point out is the wartime theory, though qualitatively correct, really misses the boat quantitatively on how many linkers that you need to get to gelation. And so we built a more resolved model to study this process, and we observed the same sort of physics that you see in the experiments in the computer model, which is that you just need a ton more linker than the naive Wertheim theory would predict to get the phase behavior that drives gelation. And the simulations explain why, and maybe you've already guessed based on the physics that I mentioned earlier, which is as you, after you've added the linkers, they'll eventually bond and start linking up colloids, but really at the end of the day, they get to sort of a linker per colloid that's actually doing the work, so the effective links per nanocrystal. And there's a bunch of wasted linker, which just turns around and does sort of a back bonding or self-linking behaviors. We fit 30, 40 nanocrystal, linkers per nanocrystal that are not contributing to the gelation process. So that explains why Wertheim theory, which neglects those kind of interactions, can not do any better than qualitative predictions, but it also gives you sort of a hint of a limitation of using symmetric linking molecules. If you want to design these systems, you may think about other strategies in terms of the linker molecule design to make those linking interactions more effective. Theory also makes a prediction that you could traverse the phase diagram back to the gel by either introducing capping agents, which would displace the linker and break apart the linked network, or you could add excess nanocrystal with no more linker. So there's two different paths that you could choose experimentally to do that are predicted by theory. Just showing you briefly that you do, in fact, if you take either one of those paths, you can go from the gel, which you can see here has very different mechanical and optical properties from the dispersion, back to the characteristics that are that you would expect from a flowing dispersion. So low viscosity, the liquid sloshes around again in the vial, and then you get the big plasmon peak that you saw in the dispersion force. So these guys are reversible by the means that are predicted by theory that they would be. So one of the aspects of this project that you probably are starting to pick up on is that a lot of the properties of these systems are going to depend not only on the concentration of the linker, but also the linker molecule's properties. And so we built a toy model to kind of explore that further. And in particular, we were looking at linking molecules 8 beads long, just as before, but that have tunable flexibility. So this is just a bead spring chain, like a Normer sort of Gress-Kramer model, so bead spring polymer, but you can tune now the persistence length to go from flexible, semi-flexible to rigid, and ask what are the consequences for the phase behavior of these systems. And this actually turns out to be quite striking. And again, we're looking at colloids in this case, the sort of canonical toy model with an octahedral arrangement of patches, so 8 patches per sphere. But what you see is on the left-hand side in the fully flexible linker case, you get phase behavior like I showed before. This is in fact that same phase diagram. But if we go to semi-flexible linkers, the phase transition goes away entirely. So there actually is no phase behavior, even when the ends become quite sticky for semi-flexible polymers. And then when you go to rigid, the phase transition reemerges in all its glory. And so there's a non-monotonic trend here. Phase behavior is a function of linker flexibility. And to try to understand that, Mike Howard, who is a postdoc in my group, derived an extension of Wertheim's theory, which accounts for these sort of self-looping interactions where a linker is allowed to do this. So it accounts for this kind of property. And what he sees in these systems is that for semi-flexible polymers of this length, as you increase the stiffness, for flexible polymers of tunable persistence, what we get is that if you start at the flexible end, the loop fraction is quite low, as shown here, but then you increase stiffness and looping peaks, and then when you get all the way to rods, they're low again. And of course, this would depend on the length of the linking molecule itself and also on the distance between patches, but for the choices that we made here, there's a very striking non-monotonic trend in how many loops are formed as a function of the stiffness of the polymer. And here we explore the consequences of that. So you can see in this plot, what I'm showing you is the end-to-end probability for the linking molecule as compared to the optimal length for forming a self loop, which is shown by this black arrow. And what you can see is as we go from fully flexible, the red curve, to the pink curve, will land in the middle on a level of flexibility that really allows for looping. And once you allow for looping from the linker's perspective, even if you add noise to the distribution of where patches are placed on the particles, the consequence is always the same, that very flexible linkers will ultimately phase separate, have the propensity to gel and phase separate, so they have very strong effective cohesive interactions between the nanocrystals. Semi-flexible cis linkers don't give rise to assembly or phase separation at all, and then rod-like linkers again promote phase separation and gelation, and you can kind of map that onto a single curve, at least qualitatively or semi-quantitatively, if you plot the structure factor versus loop fraction for this toy model system. So just an example of, in the case of sort of the linker properties, how dramatic some of the effects could be. So now I want to move on to sort of the last strategy for assembly, which we explored. This is a really well-known strategy from Micron scale colloids, which is depletion. And you can think of depletion as being sort of a limit to the linker, the sort of reentrant linker behavior that we showed before. And I'm again returning as a motivation to a system of colloids that are polystyrene in terms of their composition, but they're linked or bridged together by pan-PAM polymer molecules. So before we considered a dispersion that was flowing, but then you add linker molecules, which bridges the particles in a restricted valence kind of way, it forms a gel. If you add enough of those polymers, you'll saturate the surface, and then you'll get a reentrant dispersed phase. But once the surfaces are completely covered, then they look fairly neutral to these molecules, and now adding more polymer is just like adding a depletant, which will give rise to an osmotic force, which again drives assembly. So you can actually get two kinds of gels in these systems, bridging gels and depletion gels, which is pretty interesting. Again, this is from sort of micron scale colloids. And as I'm sure you're quite familiar with the strength of the attraction in these systems is dependent on the concentration of the secondary molecule, which is acting as a depletant when it's present in high concentration. And of course, the range of the potential is determined by the structure or the size of that depletant in solution. And so that gives rise to the possibility of tunability of the strength and also the range by modifying either the concentration of the depletant or its properties. Okay, back to the experiments. Here's an example where we're looking again at so-called ITO or indium oxide doped, tin-doped indium oxide nanocrystals, this time with PEG being added to the system. So once the ligands are stripped, these particles have a charge which would help to stabilize them. And once enough PEG has been added to move you into this sort of depletion regime on the phase diagram, turns out experimentally you get the behavior that would be predicted and has been seen from micron scale colloids, but now at the nanoscale. And here I'm showing photographs of the vial in different steps for increasing linker concentration. These are 1,000 or 1K molecular weight PEG, which is being used as the depletant. And these are Sachs profiles or the scattering that you get for these different amounts of linker. So you begin with a dispersion, then it goes to a gel, then a dispersion, And then a gel, again, in this system, it ends up crashing out at very high linker concentrations. But you get sort of the rheological and structural properties that you might expect from moving along that conceptual series between bridging to eventually depletion gels. And so I'm sort of just emphasizing that we're back to this conceptual picture that has been seen for micron scale colloids, but now it's in terms of nanoscale nanocrystals. And you can develop simple theories which can make predictions about the phase behavior of such systems. And those theories predict two types of gels, as you would see experimentally. So you get a sort of phase separation that could introduce, or that could be a driving force for a bridging gel at low polymer concentrations and a depletion gel at high concentrations. And again, they sort of qualitatively tell you the same as what you get in the experiments of these systems. They're seeing sort of the transition between a bridging gel and a depletion gel. And of course, it brings up all of the interesting questions about what are the different structural, mechanical, properties of those systems once you form them. Here, just showing you that there are differences in the IR absorption of the dispersed ITO nanocrystals, shown in gray, versus the bridging and depletion gel. So you see a depression of the peak and a broadening of the extinction. And these were sort of qualitatively predicted by simulations of those electromagnetic simulations that were carried out in this system and explained further in this PNS paper that we published in 2018. Okay, the only movie that I've got, I'll show you now. This is starting in the depletion gel and then adding nanocrystal without linker, just basically taking you down this path to show that you go from a system that was gelled to indeed a system that has a flowing liquid. And so this just speaks to the reversibility that you can get in the nanocrystal depletion gel systems. This is from Delia Mill-Iron's group, and that's our co-advised graduate student's hand, Camila, carrying out the experiments. So in general, you'd like to know if this is sort of a platform that you could use for nanocrystals of different compositions, different shapes, etc. So we had a paper that explored that in more detail, and in particular looked at the shape dependence. Here I'm just showing you one example. On the left-hand side, spheres; on the right-hand side, cubes. Of course, from the theory perspective, cubes would have stronger depletion interactions just because of the effectiveness of excluding the depletants when you bring two faces together. And so just as the theory would predict, we actually get gelation in the system with cubes at lower polymer concentrations than you would get in the system with spheres. And so there's lots more to say about these, but maybe at this point I'll just leave it to the literature and move on to the summary. I think I'm running a little bit low on time now, but I appreciate your patience. So hopefully I've presented some data that suggests that equilibrium principles can be helpful for guiding how molecular species, whether they be linkers or depletants, can be used to reversibly assemble nanocrystal gels. So their concentration, of course, is sort of the way that you provide statistical control of valence in the case of linkers, or strength of the attraction in the case of the depletion gels. And then the properties of the linkers of the depletants themselves control their size. So in the case of depletant gels, would affect the range of the interactions of the nanocrystals. And in terms of the linker gels, could give you the ability to tune structure independent of phase behavior. So maybe at this point I'll go ahead and wrap up, and thank you again for your attention, and thank the funding agencies again for the support for this work.
Thank you, Thomas, for this exciting talk. Now we have plenty of time for discussion and questions. So if you have questions, you can ask
I have
it.
a question. Well, I have several, but I have one. I mean, was a nice seminar, too much material to take in, but anyway, what is the main difference between what you call depletion gels and what in the literature is called an attractive gel, in terms of the properties, the concentration, etc., the equilibrium or non-equilibrium?
I'm not making a distinction between those two. So the attractive glasses that have been, or the gels that have been formed in the systems that also form attractive glasses, these are the same conceptually as what I'm calling the depletion gels here. So I mean, the name, I just focused on depletion to be more descriptive as a contrast to linkers, but the physics is the same.
I understand that. It was very nice to see how you can go from the dumbbells to the blinkers. But if I remember anything about the attractive gels, or even the ones that have glasses too, I mean, those are usually non-equilibrium. I mean, you cannot shift the phase diagram. So with your linkers, can you shift the phase diagram? Are these equilibrium attractive gels or non-equilibrium ones? I was lost with your...
Yeah. So thanks for that question. So I mean, short answer is we've been able to show reversibility both for the linker gels and for the depletion gels. But I think there's good theoretical reasons to believe that the linker gels are, can be equilibrium, or as equilibrium as you can hope for, because you can actually compress the phase separation to low linker to colloid ratios and down to very low colloid volume fractions. And that's, I mean, so you can access a very large range of volume fractions in those linker systems where there isn't, you don't run into the problem of the system coarsening and getting locked into, very highly non-equilibrium states. Now, in the depletion gels, we don't have as much control. We can't manipulate, as you mentioned, you can't manipulate the phase behavior in the same way there. So the only thing I can say about that is that we've been able to reverse the gels that have formed. And part of that may be due to the time scales that working at the nanoscale allows compared to micron scale colloids. So these diffuse a hell of a lot faster than micron scale colloids do. And even still, we're waiting in some cases weeks in that system to get the reversibility. So it's a good point. I think that I guess my short answer is that in the case of the linkers, I think there's good reason to believe you can get to equilibrium gels. And in the depletion driven systems, I think what we're seeing is a manifestation of just nanoscale colloids being more mobile than the micron scale systems. We do get more of the reversibility than you might expect.
Okay, so the second question was, you were showing at some stage a structure factor and how it evolved with time. Do you have any theoretical understanding of that behavior for the aggregation? In your structure factor, you had 0, weak, blah, blah, blah. And
right. We don't have a theory for predicting the evolution of the structure factor.
Of the cluster size, or of the number of fraction of bonds that link as a function of time or something? You can probably relate that to the maximum cluster size or the number of bonds that are linked.
Yeah, no, I mean, it's a good point. I mean, so just trying to understand how the structure is evolving from the simulations, at least as that gel's formed. I mean, so we published a little bit about that, and we're highly interested in this because obviously the network formed is going to dictate the underlying mechanical properties, et cetera. Yeah, so we're interested in it. We don't have a theory for doing it. We have some pretty nice models, I think, for exploring it in simulation. And then we can compare those to experimental systems through SACS and maybe get some insight into the experiments as well.
Very nice. I think there's a lot more, but I think I'll leave it to the others.
Yeah. Thanks, Margarita.
Thank you.
There are more questions. Maybe I can ask one. So, Tom, I understand that by adding linkers, you make you add extra parameter to control the phase behavior, and if you compare your system to just a patchy colloid without linkers, it's more robust, more controllable system compared to simple patches? What is the main benefits of linkers?
So yeah, it's a great question about sort of what are what are the pros and cons if you want to think about it from a practical perspective. So you have, I mean, definitely if you have the ability to make patchy colloids, then you will have much more control over the valence than the sort of statistical control that you get in the linker system. You know, it's great to have a macroscopic handle on to not have to synthesize a system with very precise number and placement of patches. So that's, you know, that's one of the reasons you might want to do it. But in terms of valence, you get much more control if you can, you know, dictate the patchy structure on a particle. But the nice thing about the linker is that in addition to having the macroscopic control of the valence and not having to synthesize precisely at the particle scale, you also can introduce flexibility of the structure through the properties of the linking molecule itself. And so there's just an opportunity for doing, you know, for making self-assembling gels with different properties. It's another handle, basically. The linker has inherent molecular properties. So it's an interesting question, you know, how much leverage that provides you. And so we're exploring that there. So the other somewhat interesting aspect of this is that if you design the microscopic interactions, then you're kind of stuck with the system that you have. And if you go with the macroscopic control, then there's a possibility of having a platform approach that can work across different nanocrystal compositions, use it for different colloids. So there's an advantage there too.
Thank you. There are more questions from
I have a question, Nico.
Yes, Maria.
Thank you, Tom, for your nice seminar. So my question is the following: you make this analogy with the patchy particle system, where you have this mixture where you approach the functionality 2, and then you make a kind of analogy with your system. But wouldn't it be the right analogy to a patchy system that has a mixture of particles with 2 and more, but where you have all the interactions between different species? And in that case, you don't have this kind of phase diagrams anymore, because when you approach very few linkers, say, your particles, your colloids that have more connections, since they don't connect to each other, they won't phase separate. I don't know if you understand what I'm saying.
Yeah. So first of all, it's great to hear from you, Jose. I can't see you, but
no, I can turn on my video. Sorry.
No, it's fine. Let's go back to the.
Yeah.
You mind if I go back to one of the?
No, it's better.
Yeah. So in this case, just as one example, and maybe this will be a helpful example. These predictions that I'm showing here for the phase diagram, the blue and the red curve, they actually are considering a two-phase system, or a mixture of linkers and colloids. You know, where the linkers have the property that the 2 ends can bind to patches on the colloids, and the colloids have these 8 patches available to them. So we've done these calculations pretty extensively for the phase behavior as a function of linker concentration, and in quite a few cases compared it to simulation. So
yeah, I understand, but what I mean is that you won't have here the same effect as Shortino got in 2006, because you don't have so if you so we started with a mixture of 3 and 2, okay, but when you have only three, you have in this case, if you have only 3, you particles with 3, they will because this is a mixture. This is also a mixture for the Shortino case. Okay, so you cannot have this 2.05, 2.1, 2.2.
Oh, right. Yeah, of course,
Yes,
right.
Of course, it's a mixture.
Yeah.
It's not what you have.
Right.
So if you have only the 3 case, only with 3 patches, you get condensation. But in the case that you are studying, when you have only the colloids, not the linkers, you don't have condensation. So there's not, I don't know if you, I mean, if you don't have, I mean, if you have only one species, one of the species, in one case you have condensation and the other case you don't have condensation. So here in Francesco's case, you have a mixture of 3 and 2, okay? If you take out the 2, you still have condensation. You have just particles with 3 and you have condensation. But in the case you are studying, if you take out the linkers, the particles with 2 patches, you don't have condensation anymore.
Right.
Okay, so that's why this analogy seems
limited. Yeah,
limited, a bit limited, I think. But okay, but it's no.
I agree.
Probably you won't have this effect of empty liquids that you clearly have theoretically here on the left. You won't have it in your system. I mean, you will have condensation or coexistence at very low densities, as you find out, but it's not. You don't go to zero density like in the left panel.
That's a great point, Jose. Yeah, I appreciate you making that. I agree with that.
Okay, sorry.
Yeah.
So this is, and then the second question is about the bridging gel. I didn't understand why it doesn't. Why you have this? At some point, it doesn't gel anymore if you put too many linkers. So
why would that happen?
Yes. Why would that happen? Why wouldn't? Is there some energy benefit of not making the linkers connecting to particles? It's just that you have too few particles, too few colloids to connect, or it's just.
There's an entropy cost to connecting particles, and there's no energy benefit, right?
Okay.
Yeah, so it ends up just being that for the same energy, you have an entropy cost for the linked case.
Okay, so the linkers don't have more, don't benefit energetically on linking to 2 colloids, different colloids. Okay.
No.
And so that's the reason.
Yeah, you can form all the possible links that you could get with enough linkers, and all of them having one free end, but then you don't have any of the entropic consequences.
Okay.
Of course, there is obviously entropic consequences for the linkers, but they're there in both cases.
Okay, thank you so much.
Yeah.
Are there more questions?
Wait a minute. I have one. I cannot resist asking Thomas because he's a chemical engineer. And, okay, so can you go to Shortino's 3+2 or whatever? The slide where you had the phase diagrams.
Yeah.
206 PRL, and where you have your next. Yeah. Well, the one where you have yours next to it.
All right. Yeah.
Okay. So Jose Maria was saying that this is a mixture, but it's not. I mean, this is a Roman mixture because there is no fractionation at all. I mean, they just assume the 2 fluids have the same concentration. And that's why they have this nice looking one single component phase diagram.
Yeah.
This is Slav, but I mean, you as a chemical engineer, you must be, I mean, because there is fractionation.
So if you go to our paper here on the,
and we see effects near the critical. Well, of course, I mean, if you look far away, this is okay, but this is not a proper phase diagram for a mixture.
Agreed. I agree. This is far from the whole story.
Yes, Margarita, but you know,
for you, because I mean, especially when you have 3 component. I worked with 3 component mixtures, and I was going crazy with the phase diagrams. So if you can show me when you have a mixture of linkers, different sizes of linkers, the 2 and the 5 or something, and the particles. So you have 3 component, and again, you make a reduction to something that looks like a single component phase diagram. I know how you do it in theory. You just basically assume the concentration is the same in the 2 phases. But what about your simulations? In your simulations, you cannot stop fractionation. So what's going on?
Yes, I mean, our simulations are in, they're in a finite size cell, right? So all we're really detecting is the stability limit of the fluid by looking at and even that approximately by looking at the structure factor. So we're not attempting to understand the phases that form from that instability.
Yeah, this is fine because this is simple. You can understand it, but it's not true.
I
So
agree.
system, I mean, and as a chemical engineer, you should know that because it's, I mean,
I'll take my, Marguerite, I'll take my lashings from you. But,
no,
but if you
no.
look at, yeah, okay.
I am a physicist. I just work too much on phase diagrams. And so each time I see something like this, I don't know what the hell you're plotting because, you know, is it pressure must be constant, for example, in your simulation, maybe in the theory. But you also, I mean, you probably have a constant pressure or something. I mean, you have too many parameters. You have too many degrees of freedom. So these are probably constant pressure cuts, but not the phase diagrams, because I bet you have the same concentration. Shortino did have the same concentration on the two phases.
Yep. So if you go back to this, I know you're a busy person, but in this JCP paper there, we show the shadow curves and calculate the compositions in the different phases theoretically. So we go beyond doing just this kind of simple spinodal calculation in this JCP paper. We don't make any attempt to do this in the simulations.
But I think the information you give is enough. It's just that it's kind of very, it's somewhat mystifying. That's all. I was not going to make this comment, but I couldn't resist. Okay, thank you very much. It was a very nice talk.
My pleasure.
So if there are no more questions, let's thank Thomas again virtually, and let's hope to see him maybe next summer or visiting us as our consultant.
Yeah, either here or there. It'd be fun.
Okay, yes.
Thanks, guys.
Okay, thank you.
Best of luck on your center.
Thank
And
you very much.
Tom.
Bye.
Yeah.
Bye.
Take care.
Bye-bye.
Bye.